25 years ago, University of Wisconsin-Madison researcher James Thompson was the first to grow pluripotent stem cells. Today, researchers use stem cells to better understand the genetic origin and progression of aging, healing, and diseases in the human body. Looking at stem cells under microscopes to study how they grow and develop is leading to breakthroughs in medical research. The following video will demonstrate some of the key steps to preparing pluripotent stem cells for research as they would be performed in a real laboratory. The first step in preparing pluripotent stem cells is to ship large quantities of frozen stem cells to the lab using dry ice and liquid nitrogen. The cells are stored in cryoprotectant, which protects them from being damaged by the liquid nitrogen. Once the stem cells are inside the lab, the first thing to do is thaw the cells in a water bath for 5 minutes at body temperature, which is 37 degrees Celsius or 98.6 degrees Fahrenheit. The cells must be thawed slowly because thawing too quickly at too high of a temperature will kill the cells.
The next step is to transfer the cells via pipette from the shipping vial to the test tube so there is more room to feed the cells with food or media. The pipette is set at 1,000 microliters, which is equal to 1 milliliter. A sterile or clean pipette tip is pushed onto the end, and then the plunger on the top is depressed until it starts to get harder to push. This increase in difficulty is called the first step. Then, the pipette is inserted into the vial and slowly releases the plunger to suck up the contents of the vial. The plunger is pushed back down to release the cells into the new vial, but the researcher must push harder than before to get past the first stop into the second. This makes sure that all the cells leave the pipette. Next, 8 milliliters of media are put into each test tube using a serological pipette. The top trigger on this larger pipette sucks up liquid and the bottom trigger pushes it out. Once the media is inside the test tubes, the tube is gently rocked back and forth about 12 times so all of the cells within the test tube will have media. Then, the test tubes will be centrifuged at a speed of 1,000 times a minute for 5 minutes. This pushes the cells to the bottom and separates them from the cryoprotectant and the media introduced earlier. Once the centrifuge is complete, you can see there's a small pellet at the bottom of the test tubes. This contains the pluripotent stem cells we want to isolate. The excess liquid, also called the supernatant, is poured off into the biowaste, and the cells are then fed with 10 milliliters of pure fresh media via the serological pipette. The last step to preparing the stem cells is plating. For our demonstration, we will use our serological pipette to transfer the cells and media from test tube to plate. You can tell if you've done a good enough job mixing or pipetting if there are no clumps on your plates. Once plated, we can actually look at our stem cells using a compound microscope. Here are some examples of what live stem cell colonies actually look like under the microscope. Stem cells are used in basic biology research, which allows us to further understand how cells function. They are also used for transplant research, to study how to replace diseased, missing, or defective cells and tissues with healthy cells and tissues that the body's immune system will not reject. Early clinical trials around the world have begun to use specialized cells grown from pluripotent stem cells to try and treat conditions such as age-related macular degeneration, heart disease, ALS, spinal cord injury, and more. Another goal is to grow whole organs such as the bladder, kidney, or heart from stem cell derivatives. There has been some success growing organs in laboratory settings, but whole organs grown from stem cells are not yet ready for human trials. The following video gives us a better idea of the potential for how stem cells could be used in treating diseases that permanently kill cells in our body, like heart attacks. These cardiomyocytes, or heart cells, were prepared at the UW-Madison from stem cells harvested from bone marrow from the legs. As you can see, they beat on their own. One of the biggest uses for stem cells is the pharmaceutical industry. Scientists grow stem cell derivatives in large quantities for testing new medications in lab dishes before moving on to animal and human testing. Stem cell research has changed science and medicine in so many ways. The next generation of stem cell researchers will make even more discoveries. Students interested in stem cell major in biology, genetics, cell and regenerative medicine, bioengineering, biotechnology, bioethics, and many other areas.